the DNA parts and replication, okay? What it says here is we need to be able to label these general places on this DNA molecule, okay? And shortly we're going to go over the differences between DNA and RNA. And one of the really clear factors of DNA is that it's double-stranded, okay? There are two halves. In addition, there is a very specific type of sugar in a DNA molecule. That's deoxyribose, okay? It's deoxygenated ribose. So the first thing I want to do is go ahead and label these parts. First, we've got the sugar, and again, I already told you that that is actually deoxyribose. The part of this diagram of DNA, which is unwound, okay, this is almost like the ladder view, that is the sugar, is this pentagon right here and below it, okay? So this is the sugar. And so are all these and these, okay? Can anyone tell me what shape the phosphate is? It's the circle. This should look a lot like the phosphate that you saw in ADP and ATP, adenosine diphosphate and adenosine triphosphate. Remember when one of those phosphate groups broke off, it released energy, and when it was bound to the adenosine, it gained energy, it stored it, okay? So this one is the phosphate. Anytime that I ask you about the backbone of the DNA molecule, I'm talking about what gives it structure, okay? What gives it strength. In a ladder, it would be the two poles that hold the rungs. In a staircase, it would almost be the railing, okay? The backbone is the phosphate and the sugar, okay? That's the backbone. And the nucleotides are just this center section, nucleotides. So what we need to do now is we need to fill in those missing bases, okay? There are a couple missing bases, but you guys need to remember that each base has its pair, its mate, okay? Just like your left and your right shoe. They're both shoes, but they're very different from one another, okay? So the first thing that I want to do is clarify this section because some people struggled with this. Do you notice how this side has two blanks? This is like a legend on a map, okay? Where one state might say, oh, that state had this many local businesses and this state has another 1,000 local businesses. What? This is all diagonal striping, though. Which letter has the same diagonal striping? A. Okay. Which one has the sideways striping? C. Okay. That's all that was. That's not supposed to be overly complicated. But now we need to go ahead and we need to fill in our complementary base pairs. Okay? So those nucleotides can find their left and right shoe. So what does A pair with? T, T, A, G, C, G, T, and G. Nice job. That's how we are able to identify and distinguish the different parts of the DNA molecule. Are we clear on that, yes or no? Awesome. So the next part is where we're actually talking about DNA replication. And before we can even talk about how RNA helps us get the DNA out of the secret layer or the nucleus, we have to actually understand how DNA is going to replicate, okay? So what I like to do first is I want to go ahead and highlight what the parent strands are, what the original strands are. And that's this side and this side, this side and this side, this side and this side, and then these are two of the originals. First thing I want to do is I want you guys to help me identify what the complementary base pairs of this are. Just go ahead and shout them out. T, T, C, G, A, T, C, C. Do you see how either side of the parent strand is different from one another, but they match up to create a pair? They're almost like puzzle pieces. Okay. So the first thing I want you guys to understand is I'm asking you to identify DNA helicase, DNA polymerase, and the replication fork. When you reach a point in a road where it splits, what do we call that? A fork, okay? So all the replication fork is where it splits. It's just where it splits. And the first thing we do is we need to unzip and we need to untwist that DNA molecule so we can actually make two separate molecules because we started with one, but we want to get two. We need to double it. So what is the enzyme called that unzips it? DNA helicase. Exactly right. Once DNA helicase has done its job and it's split those two DNA parent strands, we only have half of each pair. Okay, it's like having a left and a right shoe that are separated from each other. This next enzyme goes in and it adds bases. This is like Meghan Trainor. It's all about that base. What do we call the enzyme that adds the bases to the parent strands? DNA polymerase. Yes. DNA polymerase. Once it does that, guys, we add 2 new strands. And now what we have is 1 DNA here and 1 DNA here. These 2 separate backbones create the DNA molecule that we know. These 2 separate backbones create the DNA molecule that we know.